With this, I would now like to present to you all a brief profile of our next speaker, Dr. A. Ramasamy. Dr. A. Ramasamy obtained DPhil and Masters in Applied Statistics from University of Oxford and has an expertise in the area of specialized statistical genetics, bioinformatics, code monkey, and p-value generator. He worked as a research associate in King's College, London, honorary research associate in UCL Institute of Neurology, and also Imperial College of London. Dr. Ramasamy received various prestigious awards like Young Scientist Sponsorship from European Respiratory Society, the Top Student and Best Student Awards. Dr. Ramasamy is a member in various scientific bodies like American Society of Human Genetics, European Respiratory Society, Royal Statistical Society, and also Science Council. He is actively involved in both teaching and research at various levels. Dr. Ramasamy published more than 85 peer-reviewed research articles with citations 9,000 and H-index 38. Currently, Dr. Ramasamy has been working as the senior scientist at A-Star, Genome Institute of Singapore.
Hi.
It really is a matter of immense pleasure to have you amongst us today. So may I now request Dr. A. Ramasamy to please deliver his lecture. So please.
Let me first start my video. Hi, everyone. My name is Adai. Don't need to call me Dr. Ramasamy because that's my dad's name. Hi, so can I share my slides? I'll just share on screen one. If I make this big screen.
Yes, sir, we can see your slides.
Can you see my slides or are you seeing another screen?
We can see your slides, sir.
Okay. Sorry, I got 3 screens. I'm not sure where the hell I'm looking. Okay. Right. Okay. So I just want to quickly, I know this is lunchtime, right? So I don't want to take too much. It's not going to be a very heavy topic. It's quite lighthearted. It's just trying to walk through my career option. I mean, like my career path and see whether some of this is relevant. I mean, I understand quite, there's quite a number of students here, right? So if you all at any moment want to have any, ask any questions, just pop it in the chat or just unmute and just say something. So we can try to make this a little bit more interactive. Is that okay? Right. So first of all, thank you very much to Dr. Rangarao and the Vignan Institute for inviting me to give a talk. It's an honor and privilege to be presenting at the InSyB. I'm just going to, as I said, I'm just going to run through a few of my slides. I put the code monkey and p-value as a sort of a joke, by the way, in case you're all wondering what on earth that is. Can you see my second slide now? Oh, you're still seeing my first slide. Okay, somebody's saying chat. Yes, thank you, Nikita. Okay, so what I did for my PhD is basically I was attached to a very prestigious group on clinical trials, and who were very much into study design and how do you get the best study, right? Like what is, because this is the beginning of your experiment. A lot of the omics technology is very expensive. And the last thing you want to do is generate data and then realize it's actually not suitable because your study design was flawed. So we started with a number of techniques, which the first one we did is we try to look for sample size, right? Like we want to know what is the minimum number of sample size we need for a given project. And it turned out that actually at that time, nobody was doing the sample sizes we needed. So rather than just being very negative and saying, no, every study that you have published is underpowered, we changed the topic to say, okay, can we actually combine studies that are already published? So that's how we came up with the topic on meta-analysis for microarray data sets. Okay, so this PhD was entirely free. We took all the data that we needed from public repositories such as Gene Expression Omnibus, GEO, and Array Express. Okay, and we got a pretty good paper out of it, and the paper is more on how do you do such a study? How do you combine data sets? And there were a number of challenges, but it was very good because using free data, we were able to generate novel insights. Okay, so that's the first. So it was good experience working in a PhD, and then I decided, okay, I want to go and work in a bigger group, right? So that's how we ended up with the my first postdoctoral training, which was in King's College, which was in Imperial College, and we were working on largely on genome-wide association studies, focusing on asthma, allergy, and lung function, right? So, for example, what you can see is on the bottom, the colorful chart is what we call a Manhattan plot, where you're plotting the p-values of the genetic associations, right? And what you can see is there are several areas where there's very highly significant hits, which is the thing I circled in red. Then you can zoom into that region, and then that's what we call locus zoom plot. That means we zoom to that particular locus. We see, yes, it's not a spurious hit. There are other highly correlated SNPs in that region, which are also giving you high significance value. And then you can go back and you can look at the individual studies. So typically for a GWAS study, you need tens of thousands of samples. So depending on the phenotype, but generally you need quite a large number of studies. So then what we did is we went back and we say, okay, let's open up the studies and make sure they're all showing consistent effect. So here, this is a rather small collaboration. It's got 6 studies in it. So we confirm that the direction of association is the same and the magnitude is about the same for all 6 studies. So what we are seeing is realistic, okay? This GWAS was fantastic. It was the era, we were doing the GWAS era. There's a lot of work. And I think the fascinating thing about this is it was the first time, I believe, a lot of scientists actually openly shared data. Before that, everybody's like, okay, this is my data. I'll work on it. I'll publish. After I publish, I will share the summary results with you. But here, in this time, everybody realized if you have a sample with a GWAS cohort of 2,000, 3,000, it wasn't good enough. So we started collaborating, we started sharing data openly, and we were able to do things that we were not able to do before. So it was excellent experience for training. And for a lot of scientists as well, is how do you share data, right? And there's agreements that you have to write. So there were a lot of paperwork around it. And towards the end of my postdoc training, I started to lead or co-lead 2 or 3 GWAS consortia. And again, that was a different skill set that I had to acquire. Like, how do you manage different times, time zones, participants, and so on. Okay. Then I got a bit disillusioned after about 3 years of doing this GWAS hits, because a lot of GWAS hits are actually located in intronic regions or intergenic regions. So more than 90% of the hits we are finding do not code for a protein, right? 95% of the SNPs. Although it was fantastic and robust science, it still left something lacking. Like, you're trying to understand what is going on, right? So what we do is then I moved on to my second postdoc, which was in King's College. And this time we were looking at expression QTLs or eQTLs, right? And the main aim is, what you want to see is on the right-hand side, you see, for example, there's a transcription factor gene. You might have a mutation in that gene. And because of the mutation, You may not be able to transcribe the transcription factors, therefore you may not be able to target the genes of interest, right? So the question is, and then if you see on the bottom right, you can see AA is the wild type, and then when there's mutations, the expression levels of that particular gene goes down, okay? So we did this in 10 brain regions. We generated the exon level expression as well as the genome-wide SNPs. It was a very highly computational work. We were doing trillions of tests and then trying to get something out of it that we can explain. We got a lot of hits actually, but we found it very challenging to describe this in a paper, right? To compress that amount of information to 3,000 words to build a story, okay? So this is was quite a interesting experience, because it's not that we didn't have negative results. We had a lot of positive results, but how do you explain this, right? How do you maximize the value? So one thing we did is we started partnering with a lot of GWAS consortiums. Every time they found a SNP, we would go and look in our database and we say, right, can we explain some of your hits, right? And then eventually got to a point where there were too many requests, so we built a website and we just made it public and say, okay, if you're interested, just go and find it yourself. And the data is also publicly available. So again, you know, it's free to take and work with. Then after another 3 or 4 years, I wanted to go back to Oxford, and I got the opportunity to do so as a junior group leader. So we managed a transcription core facility. What they didn't tell me at the interview is I will be managing stuff in the wet lab as well, and I'll be responsible for processing the sample. I thought it was just the analytical skills they needed, but they needed somebody to manage the wet lab. So that was scary. The first few months was very, very scary, and I actually went into the lab to kind of understand some of the techniques. I still cannot pipe it, but I just to understand the workflow and manage the staff, right? So this was quite an exciting time. So within 3 years, we processed more than 3,000 samples. And I had another junior bioinformatician with me. So me and him, we would analyze the data coming out of this. And then we would actually say, right, this data doesn't look good. Can we go back to the wet lab and figure out what are the steps that was done? Was there any change in the reagent lots? Was the study design wrong? What's going on, right? And we also were able to do a bit of science as well, actually. So this is what we would call systems biology. So that you see the yin and yang chart on the right-hand side. So what we have is very much. So this is on the white circle. You would get the participants in. You would vaccinate them, and then after say 2 to 3 weeks, you would actually bring them back, and you would challenge them. So if it's so I was working in a vaccine institute. So if let's say it's malaria, they would fly in the mosquitoes, right, all the way from Africa, and they would deliberately infect people who were vaccinated, right? And they would take the bloods, and then from the bloods, we would generate transcriptomics data. For certain diseases like Ebola and TB, you know, obviously you can't do the deliberate infection because there's no effective treatment. Malaria, it was okay. Flu, it was okay. So certain things we could. So we had a very good idea of whether the vaccine was efficient or not. But also we were interested in the immune profiles. What are the immune profiles triggered that can indicate whether somebody would be protected or not? Now, with that information, what we would actually go back is we'll go back into the lab and we would say, right, it looks like this particular pathway is really important for immune activation, but it's not there in every human being. So can we add an extra compound that would compensate for this, right? And then Jenner Institute also had the capability to actually do the modeling, the molecule detection, and then they would do it in the small animals, and then they would be able to go back, redesign the vaccine, and go back and test it again in another set of people. So this was really fascinating because in one physical campus, one physical area, we had the vaccine manufacturing facility, we had a clinical trial facility, we had the hospital nearby. Luckily, we never had to use them. We had the labs, we had the sample storage, and then we have the analytical team, and we also had the team that built the vaccine. So all of this was nicely situated, and it was very well oiled. So they are also doing something on COVID vaccine now at the moment. So this is fascinating, right? Then as there wasn't enough vitamin D because there was no sunlight. Anyway, I was a bit homesick. So then I said, okay, let me come back to Singapore, which is close to home for me. And what I had, so that brought me back to Singapore, which is where I am. So in Singapore, I take on multiple hats. So one of my staff is tasks is to basically take care of the IT team in GIS. I know it kind of sounds a bit not very exciting, but every day there's a new challenge, and if we don't solve that challenge, the researchers will not work. And what I bring to the whole, I mean, I'm not IT trained, but what I bring to the picture is what are the needs of a scientist, right? Because sometimes the IT folks will just go on about security and all of that stuff. Kashish, maybe I'll take the question later. So Kashish is asking what kind of analysis did I do in the, so what we did is we generated data using microarrays here or RNA sequencing. So if it was animals, we would use RNA sequencing, and we would try to correlate those profiles with, let's say, T-cell responses, either through ELISA or antibody responses through ELISpot. So we're trying to correlate those two and trying to find the markers of immunogenicity. Yeah. Very challenging work because there are many things that can affect immunogenicity, including the host response. As Professor Bruno said, the antibodies, they're very challenging. Okay, so we did have some inclination, we did have some ideas about what was causing it. So some of this work is still a bit confidential, so I can't discuss too much about it. But it's fascinating work, actually. Where are we? Yeah. So and also strangely enough, when they built the COVID testing facility in A-Star in response to the pandemic, they asked me to manage the IT infrastructure. Again, there was a lot of learning to do, but I think my bioinformatics training helped me a lot. And I'll explain why in the later slides. My real passion is in trying to teach and train people. So we do a lot of introductory courses. We do classroom style, but we also have a lot of one-on-one teaching. And a lot of this has actually in the last 6 months moved online. And that was another quite interesting aspect. We had to learn how to teach online. So it's not as simple as, okay, sometimes it was simpler, sometimes it was harder to teach online. But that required, I think everybody here had to learn that as well, right? And then when I also got a small group who focuses on the scientific projects. So we focus mostly on bulk and single RNA sequencing technology, mostly 10x and so on. But we also do a little bit on RNA editing and, sorry, that should have been RNA silencing projects, okay? And we work the model as a fee for service. So we either charge our collaborators who can be internal from GIS or can be other RIs. We do charge them a fee for it. We are quite, I think, quite cheap. We're quite generous because we don't just give them the results and say, go away. We actually, even though we charge them for the analytical time, most of it is actually for computing. We use a lot of AWS computing, so a lot of that goes to cloud computing. But we actually will follow our collaborators until the end, until they get a publication, right? So in return, we ask for co-authorship on that. So although I say fee for service, I don't think that we're actually profiting very much. Actually, no profit. But sometimes they don't have the funds. So what we do is we do collaborative projects. So we say, okay, we'll work on you on the first project, we'll help you with it. And then when they apply for grants, they put us there, and then a certain amount of funds come to our core facility so I can keep my staff hired, right? So the summary of my career is actually it's highly diverse, both in terms of technology and analytical methods, disease and biological domains. So I work from anywhere from vaccine studies to pregnancy to metabolic health to editing, so it is very diverse, and it's highly diverse who I work with. Sometimes it's the IT folks, sometimes it's the professors, sometimes the students, right? And different domains as well. But what brings them all together is they are all highly computational, okay? And they're highly collaborative and highly multidisciplinary. So we are in constant communication with our collaborators. We don't work where they send us the data and then 1 year later, we just send them the paper or anything like that. So ours is a continuous collaboration. So every couple of months or every week, or sometimes depending on the nature of the project, we are in contact with that person, okay? Which is good. So now enough about me. Talked a lot about me. So as you probably know, this might be what you guys familiar with, right? Like the traditional academic pathways, right? Like first you're a student, and then when you're a student, your focus is trying to learn, trying to gain experience, trying to get technical skills, right? And then when you gradually go on to be a master's student and a PhD student, after that, you'll become a postdoc. That's a typical academic pathway. So there your focus changes to more publications, collaborations, trying to get grants, but still primarily I would say publication is what you're assessed on. And then once you're done, you think you're done, then you are like a junior PI or junior professor, and that's when the real stress begins, okay? Because now the expectations are higher. So suddenly, not just publication, but you're going to be reviewed. Your KPI is going to include grants, patents, who you know, how many students have you graduated, are you able to give lectures, and then take on admin work, right? And then later, after a few years, once you have crossed that burden, that boundary, you become a senior PI, right? And now your main duty is admin work, okay? So you can ask your professors, they will tell you how much work is in the admin work, trying to get the paperwork, trying to apply for grants. But now they're not applying grants for themselves. They're applying grants so that they can keep all the rest of us hired, right? And they also play a very important role where they're mentoring junior PIs, postdocs, and they are also responsible for managing collaborations, which basically means they also have to respond to all the funding bodies, right? So this is your traditional pathway. Okay. Any questions? In case you're not aware. Okay. So,
hello, Professor Ramasamy, sir.
Yep, go ahead.
I'm Professor Krupanidhi.
Hi.
So we are glad to have a few suggestions from you what type of the what type of the topics that our students can take up for their projects, particularly contemporary interest bioinformatics topics.
Oh, okay, yeah, I will come on to that. So I've not, I deliberately avoided anything scientific because I think there would have been plenty of scientific. I was kind of trying to get more towards the, let me do a couple more slides and then I'll come back and answer your questions. Is that okay?
Okay, thank you. Thank you.
Sure. So the traditional academic work life has a lot of advantages, okay? There's a lot of work-life balance. You know, sometimes I want to work from home, fantastic, I can do that. Sometimes I want to work on the weekends, great, I can do that as well. There's a lot of work-life balance. You have the freedom to pursue what you're really curious. You can always say, nah, I'm not interested in that. I want to work more on this. There's a lot of opportunities to travel, either in terms of presenting at conferences, but also for career-wise. Because one thing I can tell you is bioinformaticians are in high demand because a lot of the, some of the bioinformaticians actually have left to data science. That has created a vacuum both in Singapore and generally in everywhere. Because of the vacuum, there's more demand for bioinformaticians again, right? Especially junior bioinformaticians. And bioinformatics is a really good way to contribute to science and society, okay? But there are certain disadvantages, okay, about academic pathways, because the first thing is, especially in NGS technology, the technology is changing very rapidly. Every 5 to 10 years, there's a major development. So if you think about it, maybe 10, 20 years ago, microarray was hot topic. And then RNA sequencing was the hot topic. And now it's single cell technology, single cell RNA-seq. And then the future is probably going to be spatial transcriptomics, okay? And the other drawback is funding. So both either your own salary or to fund the project that you want. This is why you end up seeing people doing projects with 2 samples or 3 samples because they're very, very expensive, okay? There's also the career progression is sometimes a little bit uncertain. How do you go from a postdoc to a PI? That stage is a very challenging stage. And application for funding is also very difficult because sometimes it's 20 applications to 1 successful grant, you know. And the last point I want to highlight is loneliness. Okay, so this is a real, it's not a fluffy thing because it can be very lonely because you might be 1 or 5 or 10 people in the world who really understand what you're doing, right? And because you don't have that many, once you have reached a certain, when you talk to your professors, right, they will tell you like, oh, I wish more people understood what I was doing, right? When you reach the top, it can be a bit lonely, right? So that is some of the stuff that pros and cons of academic pathway. But if you look at the common skill sets, right, like what are the skill sets that is required for a bioinformatician? The first and foremost is computational skills. Now, bioinformatics itself has many different meanings. It could be protein bioinformatics. It could be next generation sequence bioinformatics. It could be imaging bioinformatics. So there are lots of different bioinformatics, but at the core of it is computational skills, right? So get really good at your computational skills. R, Python, those are the 2 leading programs. So get really good at your skill sets, okay? The other one is data literacy. So how good are you at looking at the data and finding outliers? Can you look at a data and say, look, certain things are rounded to 0.5 and 0.25, but then once in a while I see a 0.367. So what, you know, that, are you able to look at the data and spot those anomalies, right? And part of the data literacy it comes through both analysis and coding skills. Then you have mathematics, okay? If you don't like mathematics, please try to learn a little bit because it will take you a long way down. So especially algorithm development and probabilities. So these are very crucial skill set. But there is another aspect which is slightly forgotten, what makes a bioinformatician really good. Communication skills, because you're always talking to biologists, non-technical people, sometimes even among technical people, you still got to explain to them. You got to present your ideas, either in the form of presentation, writing, data visualization. So these are all quite valuable skill sets, right? And where I'm just giving you some examples of where you might want to think about going forward, right? If academic career are not for you, what are the non-traditional pathways, right? So as I said, like managing core facilities, because usually bioinformatics folks are very organized, even from the way you organize your data, your scripts, you're very organized. You're much more organized than most people, right? So managing core facility is not, it does require you to learn a few things, but you can do that. Bioinformatics as a service. I know there are a number of companies in India and elsewhere that actually do bioinformatics as a service. Collaborators. Programming skills, data visualization, teaching, and of course, ultimately, data science as well, right? I mean, this is why a lot of bioinformaticians actually ended up in data science, because they have built years and years of experience working with data, understanding data, and how to communicate data, right? So that's how they ended up with being data science. So don't just look to universities and research institutes, but look into hospitals, pharma companies, software companies, even journals. A friend of mine was recently just hired into a patent search company. So they're actually mining patent data to look for which patents are worth funding or purchasing further. So they have taken up quite a number of things. I have a few more slides, but I want to answer the question, what kind of topics and projects can your students take on, right?
Yeah, please, sir.
Yeah, so it's a bit challenging because a lot of this depends on data availability, right?
Yeah.
Yeah. So either you can go down the route of analytics. So that means you partner with somebody who has the data. They give you the data set, you analyze for them, and you try to find novel insights. Okay, that's one route. So for this, you really need to have strong collaborations with people who have the data, right? And the other option you have is you can, if you want to go down, let's say the algorithm route, you can do what I did, which is basically try to find publicly available data sets and try to mine for valuable insights. Now, I know I kind of say it's a free way of doing a PhD, right? But actually, it gave me a lot of useful things because you're reusing data and you actually, the results you find in one data set, if you cannot reproduce in another data set, then that result is not valid, right? So you're actually building robustness into your data. There are a couple of websites you can look into for single cell. So single cell is a very hot topic at the moment because the cost of doing single cell has come down to a more affordable price. But you can also do bulk RNA-seq. So these are the two things that I would recommend. What are the backgrounds of your students? Are they mostly master's, PhD students?
Our students are with the BTech Bioinformatics, sir, with a lot of inputs on information technology tools like Python, Java, and all those things.
Perfect. Yeah. So Python is great. I would recommend looking into R as well, because R has a lot of what do you call biological packages built in. So Bioconductor, for example, is a website you can go to. It ultimately you have to ask the question why, right? So if you want, is it just, if it's for algorithm development, no problem. There are lots of data you can download, try your method and find that.
Algorithm development, is it computational, what is it called, languages or programs need to be used, or already existing softwares need to be adapted, sir?
No, you have to come up with the algorithm, right? So like the single cell RNA-seq, one of the challenges is for cell identity. So you can do as a fancy, like it's a little bit like what Bruno also described earlier, right? So that's also algorithm development, right? So you got to understand the problem well enough, and then you got to say, can I tackle it in a different way?
Yes, sir.
Right?
Yes, sir.
Either using machine learning or AI techniques and so on.
Yes, sir.
Yeah.
Thanks. Thank you. We'll be in contact with you.
No problem. I mean, we can brainstorm later.
Yes.
So I don't know, I feel like we're all here talking about some of the important things I learned, right? Like, I think it starts with saying I'm worthy to contribute, right? I'm worthy to speak my thoughts, to listen to others, to make a change, to teach others, right? So you got to build, for students, I'm talking to students here, right? Like you got to build your presence. So I find a lot of students tend to be very shy, very quiet, and then they say, oh, I'm not good enough. So that's usually the first challenge I have with the students, is to say, nope, you're good enough, go and do this, right? So for the students, this is the first thing, right? The second thing is you can think big, but you need to do some action now. Don't say, I want to go solve cancer. Great, that's fantastic, but how are you going to do that? Break it into actionable plans. Each plan should have a aim and a goal. So that means when you have reached that goal, you can, you know that you have achieved that. So again, use your mentors, lecturers, teachers who are there to help you plan these. You have to make it into manageable chunks. Okay. And who's the best person to lead? Is it you? Is it somebody else? Right, you want to solve something ambitious, great. But if you don't have the skill sets to do that by yourself, then go and find somebody you can contribute to, right? You gotta share, and don't be afraid to fail. And especially when you fail, share the failure, right? Because if you don't share, somebody else will fail doing exactly what you did. So you wanna help others as well. You want to do something worthy, stay current. You want to find out what are the topics of interest. I think this is the question you asked me. Talk to people in the field and think about implementation, right? How are you going to come out? So the last statement there, how do you get up in the morning, right? This is my question. Like if I get up in the morning and I'm excited to go work, that means I got something interesting to do. If I get up in the morning and I go, you know what? I wish I'm going to call in sick. That means I'm not doing something useful. That means I need to go and take some action. Okay, so that's why I say, how do you get up in the morning? Know your purpose, right? Why are you doing bioinformatics? Or why is it to win something? Are you to win some prestige, to win accolades, to win acknowledgement, which is fine. We do need people who are competitive and who want to win. Or are you doing it purely out of curiosity because you want to discover new things? For me, it's because I want to help other people do better research, right? So when I was playing the PI role, in the PI role, I was expected to be more competitive, to go get grants, to win, compete people, publish before others do, which, I didn't do very well, but that wasn't very satisfactory because I was in the wrong role. So when I moved to a more support role, I'm way happier. Okay, my salary is exactly the same, but I'm way, way happier than being in the PI role to compete. Okay, nothing wrong with either roles. All roles are great. No, I don't work with plant-based data, sorry. Okay. I think the last slide I have is, no, 2 slides. Communication is key. So you need to be listening as well as speaking, right? It took me many years to actually start listening to people because I thought communication means you just talk, right? And sometimes people are complaining to you or they're saying something, but you got to understand why they're doing that, right? So that's another skill set to have. The third point is a bit controversial. Manage your boss, okay? So if you come from a very hierarchical society, you might be scared to tell your boss off that they're in the wrong direction or that they're giving you too much or too little. But you got to understand your boss. Your boss has a boss, and his boss has a boss. So the instruction is flowing down, and what are they trying to deliver? And work with them. And being a good follower is just as important as being a good boss, right? So that's what I mean by manage your boss. So in the sense that understand your boss, see what their priorities are, and try to explain what your priorities are, okay? It's not an easy skill set, but it will take time. And then the last slide I have is that the teamwork is crucial. So I think there's this famous saying, right? If you want to go fast, go alone. If you want to go far, go in teams. So I don't know who actually said that. But my first postdoc boss, Debbie Jarvis, who's amazing. She's got like lots of collaborations. And I was a very hot-headed postdoc. I was kind of like always telling people like, oh, you're wrong. You know, you should be using this program. This is not right. And she sat me down and said, look, if you want to work on your own, that's fine. You're going to be 100% right or 100% wrong. But when you work in a team, you got to be prepared to be a little bit wrong, right? So, and that's where it really opened up my eyes. And you say, okay. So it's not about being right all the time. It's about working with people, trying to find that common ground, right? So this is probably the best advice I got, and I'm trying to share that with you all. Okay, and I think that's it. Yeah, that's the last slide I have from here. Maybe contact me later about like potential projects and stuff. I can try to maybe have a chat later. Okay, thank you. Any questions so far or?
Thank you very much, sir. That was very inquisitive.